And then cool. Hi, everyone. My name is Pavitra. I'm really excited to have the chance to talk to you all today. So as biologists, we're generally interested in sort of the diversity we see across the tree of life. And one particularly fascinating aspect of that diversity is the diversity we see between the sexes, in traits ranging from morphology to physiology to behavior. My research program seeks to understand sort of the causes and the consequences of this diversity between the sexes, focusing on three key evolutionary phenomena. The first of these, sexual antagonism, refers to the fact that males and females often have different fitness optima for a variety of different phenotypes. But because males and females within the same species share the majority of their genome, they're often constrained from evolving to these separate optima. The second, sexual selection, describes a variety of different ways in which males and females within a population may interact, choose mates, and thereby determine the genetic composition of the next generation. And finally, the third phenomenon, sex determination, refers to the different processes through which males and females may or may not be sent down different developmental pathways. Now, I've described each of these separately, but of course they interact in a variety of different ways. And upstream of this trifecta, there are even more basic questions to be asked. How did meiosis evolve in the first place? What factors lead to the maintenance of genetic recombination? How and why do we see the evolution of separate sexes? These three evolutionary phenomena in turn affect a wide variety of other evolutionary processes, such as the origin and maintenance of species, the enhancement and reduction of sexual dimorphism, or the evolution of divergent mating systems. My work lies up and down this axis of sex and its consequences, from theoretical work looking at the evolution of asexuality in flowering plants, to empirical work looking at the evolution of sexual dimorphism and divergent mating systems in frogs and lizards. The majority of my work focuses on these three key evolutionary phenomena of sex determination, sexual antagonism, and sexual selection. My goal with my research program is to build new theory using mathematical modeling and simulations, and use the intuition we gain from that theory to make predictions that we can then test across micro and macro evolutionary scales. Today, I'm going to be describing a project in the branch of my research that deals with sexual selection, and in particular, the impact of sexual selection on the origin and maintenance of species. Now, sexual selection has been an important topic in evolutionary biology since its foundation. And one major takeaway that has come out of this extensive body of theoretical and empirical work is that sexual selection can be a rapid and powerful force driving the evolution of preferences and traits within populations. For this reason, we often see rapid divergence in systems of sexual selection between populations, whether those systems are based on things like sperm-egg protein recognition or pheromone production and detection, or divergence in preferences for color cues or acoustic signaling. Now, this rapid divergence of sexual selection systems between populations obviously leads to questions about the role of sexual selection in the origin and maintenance of species. And this is a really exciting time to be thinking about these questions, because over the past 10 years or so, there's been this growing realization that hybridization and gene flow are extremely common phenomena across the genomes of many different organisms. And so the big question is, well, if there's all this gene flow going on, how do species maintain distinct genomes? Now, the barriers to gene flow have traditionally been classified into pre- and post-zygotic barriers. Pre-zygotic barriers include things like spatial or temporal isolation between species, mate choice for conspecifics, and gametic incompatibilities, whereas post-zygotic barriers include things like reduced hybrid fertility or viability. Today, I'm going to be focusing on mate choice, which has traditionally been classified as a pre-zygotic barrier, because mate choice for conspecifics can prevent the initial formation of hybrids. And in a sense, it can also be seen as a post-zygotic barrier, because hybrids once formed might be disfavored by individuals from either parental species when it comes to matings. But what I'm going to do today is describe to you a further way in which mate choice can enhance any kind of post-zygotic selection against introgressed ancestry. Now, we've known for a long time that introgressed ancestry tends to be deleterious in its recipient species. But recent genomic evidence has shown us that selection against introgressed ancestry is occurring at very many loci scattered throughout the genome. From an application of Fisher's fundamental theorem, we can therefore say that the rate at which selection acts against that introgressed ancestry will be proportional to the variance in introgressed ancestry within a population. Where does that variance in introgressed ancestry come from? Well, we can take the simple example here, in which we're assuming some entirely red species 2 individuals are entering a gray species 1 population. The processes of recombination and independent assortment of chromosomes will break up and spread out that introgressed ancestry, such that in a few generations you have variance among individuals in how much introgressed ancestry they have. Now, this variance is generated both by the fact that some individuals have introgressed ancestry and some don't, and the fact that among individuals who have introgressed ancestry, you might have different amounts. Assuming random mating, these processes of recombination and random assortment will take this introgressed ancestry and spread it out more and more evenly across more and more individuals in the population, slowing down the rate at which selection can act against that introgressed ancestry. But we know individuals don't mate randomly. Most individuals will tend to prefer to mate with another individual from their same species, or at least another individual who shares the majority of their ancestry. This type of assortative mating brings like ancestry together in conjunction with like ancestry, bundling it up and concentrating it in fewer individuals in subsequent generations, thereby increasing the variance in introgressed ancestry and allowing selection to act more efficiently against it. So our goal with this project was to understand this bundling effect of assortative mating, the way assortative mating concentrates introgressed ancestry together and thereby facilitates any type of selection acting against that introgressed ancestry. We wanted to first characterize it and figure out if it was real, get at the genetic mechanisms that underlie this effect, and then go out and measure the impact of this effect in real populations. So to start investigating this effect, we constructed a population genetic model. And we're going to be considering the scenario in which there's an introgression pulse. So some entirely species 2 individuals are entering a species 1 population. We're going to assume that species 2 ancestry is deleterious, and I'll get into specific reasons why that might be the case a little later on. And we're initially going to assume a genome composed of 1,000 unlinked autosomal loci. And again, we'll relax this genomic assumption in a little bit. Finally, we're initially going to assume that mate choice is generated by a model of that assortative mating is generated by a model of active mate choice. And in particular, we're going to initially assume a classic fixed relative preference model of mate choice. Now, all this means is that if I'm an entirely species 1 individual, I would most prefer to mate with a species 1 individual, and vice versa if I'm species 2. If I'm a 50-50 hybrid, I'm going to practice indiscriminate mate choice. Now, this is one model of mate choice, and I'll present results from alternative models a little later on. Okay, so we can go ahead and track the fraction of introgressed ancestry in the population after the introgression pulse has occurred. So on the y-axis, you have the total introgressed fraction in the population shown on the log scale, and on the x-axis, you have the generations after admixture has happened. And we can look at how the introgressed fraction changes in the scenario in which there's no mate choice for conspecifics, and the scenario in which there is. And as specific, in the case where you have mate choice for individuals of similar ancestry, the purging of introgressed ancestry is far more profound than in the case you don't. But this case of mate choice is actually concealing two distinct effects. There's sort of the direct sexual selection effect of mate choice. Individuals who have a lot of introgressed ancestry are going to get fewer matings, produce fewer offspring than individuals who have less introgressed ancestry. But then there's also the indirect bundling effect of mate choice. Introgressed ancestry is going to get bundled up as a result of assortative mating, making it easier for both natural and sexual selection to act against that introgressed ancestry. So if we wanted to understand the second indirect effect of mate choice, we had to figure out a way to decompose these two effects. And the way we did that was by figuring out a way to turn off the bundling effect of mate choice. To do this, we took advantage of the flexibility of evolutionary simulations. What we did was go through every individual in the population and calculate, based on their ancestry and the distribution of other individuals in the population, how many matings they were likely to get, that is, their fertility fitness. We then took this fertility fitness and reassigned it to their viability fitness, and then allowed mating to proceed randomly. So what this is doing is that maintaining the direct fitness effect of mate choice. If you were an individual who was very attractive and you were going to get lots of matings, you're still getting a fitness boost as a result of that. But because we're allowing actual mating to proceed randomly, we've eliminated the bundling effect of mate choice. And so we can return to these results looking at the case where there's no mate choice and mate choice with a bundling effect in operation, and look at what happens when we turn off the bundling effect. And you can see immediately that the bundling effect is contributing substantially to the purging of introgressed ancestry from this population. Now, another way to look at this is to directly track the variance in introgressed ancestry within this population over time. And we can go further and acknowledge that this variance in introgressed ancestry is going to depend partially on the fraction of introgressed ancestry in the population, and sort of normalize for differences in that fraction. When we do this, we see as expected, in the two scenarios in which there's no bundling effect, the variance in introgressed ancestry is decaying rapidly. Whereas in the scenario where there's mate choice with the bundling effect, this bundling effect is maintaining ancestry variance for longer, therefore allowing selection to act more efficiently against introgressed ancestry for a longer time period. So with this, we sort of established that, okay, the bundling effect is real and it seems to be playing an important role in the purging of introgressed ancestry. But we also really wanted to dig into the genetic mechanisms that underlie this bundling effect of assortative mating. And to do this, we turned to work done in a slightly simpler setting of two locus fischeri sexual selection. In this previous work, we had identified two distinct mechanisms, or two distinct channels, through which sexual selection occurs. And I'm going to present those two channels to you in the context of assortative mating now. So the first mechanism, the first channel, we call the trans channel. Now, all this means is that a female with a lot of introgressed ancestry is disproportionately likely to mate with a male with a lot of introgressed ancestry, meaning that their resulting offspring will have a lot of introgressed ancestry on both the maternal and paternal copies of their genome. That is, there's a positive covariance in trans across the maternal and paternal copies of the genome in terms of how much introgressed ancestry you have. Now, this channel is simply a reflection of the action of assortative mating in the previous generation. But once this positive trans covariance is in existence, the cis channel can begin to operate. Now, the cis channel can be facilitated through many different mechanisms, but the simplest of which is recombination between maternal and paternal copies of the genome. This recombination will disproportionately place introgressed ancestry along a single copy of the genome. That is, it'll generate linkage disequilibrium along a single maternal or paternal copy. And so these sort of two mechanisms of sexual selection are really describing the two ways in which the bundling effect is bringing like ancestry together with like ancestry. In trans, across maternal and paternal copies of the genome, and in cis, along a single copy of the genome. We can go further and decompose the variance in introgressed ancestry in a population precisely into these two channels. So the variance in introgressed ancestry will be due partially to the variance in introgressed ancestry at particular loci in the genome. It will also be due partially to a positive trans covariance between loci across maternal and paternal copies of the genome. And it'll be due to a positive cis covariance between loci along either maternal or paternal copies of the genome. We can go ahead and also track these different covariances directly in our simulations. So if we look first at the trans covariance, remember that I mentioned entirely species 2 individuals are entering a species 1 population. So trans covariance starts off as complete. But in the two cases in which there's no bundling effect, in blue and in yellow, that trans covariance decays immediately. Whereas in the case where we allow assortative mating with the bundling effect, that trans covariance is maintained for longer through the action of assortative mating. Because this trans covariance is maintained for longer, we can also see a change in the cis covariance. Again, introgressed DNA always enters a population in full cis linkage disequilibrium. And the processes of recombination will sort of break up and sort of decay that cis covariance over time. But in the case where we allow the bundling effect to operate, the conversion of trans covariance into cis covariance is maintaining cis covariance for longer, causing it to decay more slowly. And so overall, these two channels are describing mechanistically how ancestry variance is maintained for longer due to assortative mating. So this sort of describes mechanistically what's going on under the hood with the bundling effect. But we also wanted to understand when it was important, that is, what sort of evolutionary scenarios it could play a role in. And so we essentially went out and tested a wide variety of models and evolutionary cases to see how robust the bundling effect was to these different situations. So one of the first things we experimented with was understanding the importance of the bundling effect using different models of assortative mating. So far, I've shown you results using a sort of fixed relative preference model of assortative mating. Another very common and very important model of assortative mating is what we call like with like mating. So in this case, if I'm species 1, I'd still most prefer to mate with a species 1 individual, but 50-50 hybrids would most prefer to mate with a 50-50 hybrid. A 70-30 hybrid would most prefer to mate with a 70-30 hybrid, and so on. Another model of assortative mating that's been shown to be important in the speciation literature is that of parental imprinting. So this is a case where an individual would most prefer to mate with another individual whose ancestry fraction matches that of their parents. So we ran identical simulations using these different models of mate choice. And here are the main mating models on the left, like with like matings in the middle, and parental imprinting is on the right. And you can see that the bundling effect is playing an important role in all three of these different cases. One particularly interesting effect is that the bundling effect has a particularly big impact in the scenario of like with like mating. And this is because this particular model of mate choice generates assortative mating across the entire ancestry fraction spectrum. And so it generates a very strong correlation in ancestry fraction among mates, which translates to a very strong transcovariance, thereby facilitating the action of the bundling effect. Now, one sort of takeaway from this is that it doesn't matter sort of which precise model of mate choice we're assuming here. And in fact, we happen to assume a model of female preference for male traits, but of course, as Rachel just talked about, that isn't always the case. And these models are pretty agnostic, or completely agnostic as to which sex is choosy. But taking a step back further, the core of the bundling effect is assortative mating. And we've chosen to model assortative mating as coming out of active mate choice. But in some systems, it could come out of, say, host pollinator preferences, or temporal or spatial microstructure. And all of those would work in the same way. The core, again, as long as assortative mating is generated, the bundling effect will have an impact. We're also interested in investigating the impact of the bundling effect across different genomic architectures. And in particular, one we were interested in looking at is what would happen under realistic genome-wide recombination rates, which have been shown to play a large role in the purging of introgressed ancestry. So to do this, we ran the same simulations, but using a high recombination species for which we used the linkage map of humans, and a low recombination species, for which we used the linkage map of Drosophila melanogaster. Now, if you look across these two cases, you can see that in the low recombination species, the purging of introgressed ancestry is more rapid and more profound than in the high recombination species. And this is a previously characterized effect that we've tried to dive into more in some related work. But the effect I'd like to draw to your attention to now is that the bundling effect seems to be much stronger in the high recombination than in the low recombination species. Now, this is because the conversion of trans covariance to cis covariance is primarily occurring through recombination between maternal and paternal copies of the genome. In high recombination species, the conversion of trans to cis is sort of facilitated by the high recombination rate throughout the genome. And so high recombination species end up having a stronger cis channel of the bundling effect, and therefore a stronger bundling effect overall. We're also interested in investigating how different architectures of the female preference and male trait might play a role here. In the simulations I've shown you so far, we assume the same loci underlay the preference and the trait. We sort of relax that assumption in many different ways. We assume different loci underlay the preference and the trait. We assume different numbers of loci were controlling each. And we found that almost regardless of the precise nature of the architecture underlying preference and trait, the bundling effect played an important role as long as both were sort of sufficiently polygenic. And finally, we wanted to make sure the bundling effect still operated if we included sort of a realistic genome with sex chromosomes, as occurs in many species in which we're interested. And we found that it does sort of operate consistently in this scenario. Finally, we wanted to investigate how robust the bundling effect was to different types of post-zygotic selection against introgressed ancestry. And we found that the bundling effect operates sort of almost regardless of what the precise reason for selection against introgressed ancestry is. So far, I've been pretty agnostic about why introgressed ancestry is deleterious, but there are lots of specific reasons why that might be the case. One is simply local adaptation. Species 2 genes might be maladaptive to the species 1 environment, and so introgressed DNA from species 2 is selected against in species 1. Another possibility is genetic load, and this is something that's gotten a lot of attention in the context of Neanderthal-human introgression. In this case, species 2, due to various demographic reasons, has accumulated more deleterious mutations than species 1. And so species 2 ancestry is selected against when it introgresses into species 1. Finally, it's possible that species 2 ancestry is not intrinsically deleterious, but is simply selected against when it introgresses due to Dobzhansky-Muller incompatibilities throughout the genome with species 1. I'm not going to dive too much into these results, but we did explicitly examine all of these different scenarios and found that the bundling effect increases the purging of introgressed ancestry regardless of the precise nature of post-zygotic selection against that introgressed ancestry. So with all this in mind, we were really excited to, once we had established the bundling effect was real, we had gotten at its mechanisms, we'd shown it applies to a variety of evolutionary scenarios, we were really excited to go out and measure the strength of the bundling effect in real populations. And in order to do this, we had to figure out a way to estimate how much the bundling effect increases the purging of introgressed ancestry. So we developed new methods to estimate this impact. And our methods were based around rho, the correlation coefficient of ancestry within mating pairs. Now, this is a very common and a very general measure of the strength of assortative mating. If we consider what happens after a single generation of assortative mating, the correlation between mates in ancestry is immediately translated into the trans-covariance in introgressed ancestry in the next generation. And so we can very naturally calculate how much more purging occurs due to a single generation of assortative mating. And we find out that the increase in purging increases by a factor of 1 plus rho. We can also think about what happens when assortative mating happens across many generations. So in this case, the correlation between mates is going to be translated into transcovariance. That transcovariance is going to be converted into ciscovariance. And so the calculation gets a little bit more complex. But we can still develop a method to calculate this and say that after multiple generations of assortative mating, the overall increase in purging due to the bundling effect is increased by a factor of 1 over 1 minus rho. Now, this latter calculation depends on slightly more assumptions about the strength of selection and the change in variance in introgressed ancestry. But these two can be taken very conservatively as a lower and an upper bound on how much the bundling effect increases the purging of introgressed ancestry. And so we were able to take these methods and sort of go out and apply them to real hybrid zones. And the first hybrid zone we considered is the hybrid zone between the yellow baboon and the Anubis baboon in the Amboseli Basin in Kenya. And this work is sort of done as part of the Amboseli Baboon Research Project. And before I go further, I just want to really thank Jenny Tung and her PhD student Ariel Fogel for sharing their data with us and for a lot of really wonderful discussions. So in this particular case, Anubis baboon ancestry has introgressed into the yellow baboon population. And previous genetic work from Jenny Tung has shown that Anubis baboon ancestry is on the whole selected against in the yellow baboon population. Now, the Amboseli Baboon Research Project is an example of really phenomenal fieldwork. And so we basically know who mated with who in these populations. And so and thanks to Jenny Tung's lab, we also have precise estimates of the genomic ancestry from all these individuals. So we can take this data and actually just go in and directly calculate the correlation between mates in introgressed ancestry. And we get this correlation of approximately 0.2, which, plugging into our calculations, means that there's 20 to 25% more purging of Anubis ancestry in this population due to the bundling effect. Now, this is also a nice illustration of the utility of our method. These baboons live in a very complex social structure. They move around in social groups. There's dominance rankings in both males and females. It's all very complicated. But by using sort of this metric rho, we can kind of get at how much assortative mating actually did take place, sort of regardless of the complex social structure which surrounds these individuals. Now, this is a case of relatively weak pre-mating isolation. The next case I'm going to show you is one of strong pre-mating isolation. And this is the hybrid zone between Xiphophorus cortezi and Xiphophorus birchmanni in tributaries of the Rio Santa Cruz in northern Hidalgo, Mexico. Now, and I should say that I really want to thank Molly Schumer and our postdoc Dan Powell, who shared this data with us and also discussed it extensively. So in this particular hybrid population that we're going to consider, Xiphophorus cortezi is the major ancestry, and Xiphophorus birchmanni is the minor ancestry. And consistent with what we see in other swordtail species, birchmanni ancestry is being consistently selected against at very many loci throughout the genome. Now, in this particular case, we don't actually have the ancestry correlation in mating pairs. Instead, Molly and Dan very carefully characterize the ancestry fraction of mothers and the resulting offspring from this population, which we can turn in. Which we can back calculate the correlation between mating pairs. To be precise, we're using the regression of offspring ancestry onto mother's ancestry to get at rho the correlation between the mating pairs in the previous generation. So if we consider, say, this point up here, mother's cortezi ancestry is on the X, offspring on the Y. If we consider that point, that's a mother who had a lot of cortezi ancestry, and she had an offspring with a lot of cortezi ancestry. So that female presumably mated with a male who had a lot of Cortez-I ancestry. Instead, if we consider this point down here, that's a mother with a lot of Cortez-I ancestry who has an offspring with much less Cortez-I ancestry. So that's a female who very likely mated with a male with a substantial amount of Birchman-I ancestry. And so we can transform this calculation sort of back into the correlation between mates, which we get this very high value of 0.93. And plugging that into our calculations, we see that this means that there's 2 to 14 times more purging of introgressed ancestry due to the bundling effect of assortative mating in the system. Now this system of like strong premating isolation is an interesting one to think about. So this is a case, as I said, where premating isolation is strong. Introgression is rarely going to happen. But when it happens, that introgressed ancestry is going to be purged rapidly and efficiently. And that purging is going to be very much enhanced by the action of the bundling effect. So I hope I've convinced you somewhat, using both these theoretical and empirical results, that the bundling effect contributes significantly to the genetic isolation of species. We're very excited about this work, and we're also very excited to extend it even further. A couple future directions we're excited about are looking at the impact of the bundling effect of assortative mating on situations in which there's continual gene flow, that is stable hybrid zones. And in particular, trying to investigate how the bundling effect impacts cline width and cline stability. Another direction we're pursuing is trying to use our mechanistic understanding of assortative mating and sexual selection to really go after the genomic signature of assortative mating in hybrid zones and see whether we can recover some sort of consistent genomic signal of this process. And potentially going even further to see whether we can transform that into getting a consistent genomic signal of assortative mating within population, not based on ancestry. So this has been sort of a quick overview of the branch of my research that deals with sexual selection. And generally, I'm just really excited to continue investigating what I see as these 3 really key evolutionary phenomena of sexual antagonism, sexual selection, and sex determination, and their impact on patterns of genetic and phenotypic variation and adaptation, both within and among species. I'm also very excited to announce that I'll be starting my own lab in fall 2023 in the Department of Ecology and Evolution at the University of Chicago. So if anything that I've said is interesting to you, or you think it might be interesting to you, please come find me. I'd love to talk more, or shoot me an email. And with that, I'd just like to thank my wonderful co-authors, Graham Coop and Carl Veller, all the people who have shared data and really nice discussions with us, my funding sources, and the American Society of Naturalists for inviting me to be here today. And thank you all for listening. I'd be very happy to take any questions.
I should keep my mask on. That was a very cool talk. So I work on DMI, so it's kind of a Dobzhansky-Miller incompatibility question. There's kind of 2 ways to think about DMIs on sort of extremes. One is sort of like the quantitative genetic way, like Fisher's geometric model way of sort of co-adapted genomes where everything interacts with everything. And then there's sort of the discrete pairwise model. Could you elaborate on which or both, or like how you implemented DMIs and how that might affect things? Because the quantitative genetic model kind of has its own sort of bundling thing sort of built in.
Yeah, no, that's a great question. So we went with more of a pairwise approach. That is, we took a genome and essentially scattered loci along it that were interacting. And I sort of simplifies the DMI results. To a certain extent, say selection against introgressed ancestry due to DMIs is really frequency dependent. And so we actually also looked at simulations in which we started off the population with 50-50 of each ancestry. And in those cases, the bundling effect actually initially prevents the purging of either type of ancestry because it's holding genomes together, essentially. Once one ancestry starts to decline, however, it steps in and really starts to enhance that sort of selection against introgressed ancestry. So it is a little bit more complex. I think that's all, but thank you.